All right, let's dive in. Today, we're going to unlock the secrets of some of nature's toughest survivors. And we're going to do it by looking at the really cool staining technique that makes them visible in the first place.
Okay, think about this for a second. How on earth can some bacteria survive being boiled in water, or blasted with disinfectants, or even the vacuum of space? I mean, that's wild. It's like they have the superpower, right? So what is their secret weapon?
Well, that secret weapon is something called an endospore. And get this, it's not for making more bacteria. Nope, it's a survival pod. It's basically microscopic body armor that a bacterium builds around itself when the going gets tough.
But here's the catch. That amazing body armor, it's a huge pain for microbiologists. See, its coat is made of keratin. Yeah, the same stuff as your hair and nails. And it acts like a little raincoat for stains, just making them bead up and roll right off. So if you try a normal stain, poof, the endospores are completely invisible.
So how do we see something that's literally invisible to our usual methods? Well, scientists got clever. They came up with a workaround based on a pretty simple idea. If you can't get through the gate, you force it open.
And this is where it gets really cool. Look at the difference here. Standard staining, done at room temp, is totally useless. It just bounces off. But the special endospore stain, called the Schaeffer-Fulton method, adds one crucial ingredient: heat. You apply the stain with steam, and that heat forces that tough coat to expand just enough for the dye to sleek inside. Brilliant, right?
Okay, so that's the principle. Now let's walk through how it's actually done. It's a really elegant process, a few simple steps that let us color the endospore and the regular cell completely different colors.
So step 1, you flood your sample with a green stain, malachite green, and you hit it with steam. That's the part where you force open the gates. Step 2, you just rinse it with water. It's gentle, but it's enough to wash the green stain out of everything except those tough little endospores. Then for step 3, you add a red stain, safranin. This colors all the regular bacterial cells that are now clear. And finally, step 4, you look under the microscope.
And this is the payoff. What you see is, well, it's not just beautiful, it's incredibly informative. That color contrast tells you everything. It's super simple. The endospores, the ones that stubbornly held onto that first green stain because of the heat, they're green. The regular active bacterial cells, we call them vegetative cells, they show up as red or pink from that second stain. You absolutely can't miss it.
And yeah, this quote just nails it. You see these bright green ovals, and they can either be inside the red cells, like they're getting ready, or floating around on their own. It's just a crystal clear confirmation that they're there.
Okay, so this is way more than just a neat lab trick. This is a critical tool for public health and safety. Seriously, knowing how to spot these things can have life or death consequences.
For example, in medicine, you use it to identify the bacteria that cause nasty diseases like anthrax and tetanus. In hospitals, it's crucial for checking that sterilization equipment, autoclaves and stuff, is actually working in killing these super tough spores. And, in food safety, it's essential for making sure canned foods are processed properly to kill things like Clostridium botulinum, which causes botulism. And just to give you an idea of how tough we're talking, an autoclave, which is basically a souped-up pressure cooker, has to get up to 121 degrees Celsius. That's way above boiling. And it needs high pressure on top of that just to be sure these things are destroyed.